Can AI extract the story of cancer care? Evaluating LLaMA 3.1 for clinical entity extraction in breast cancer records. My name's Sam McInerney, and this work was supported by Mattis Fallis, Franz Gruber, Arlene Casey, David Lowe, and Peter Hall at the Universities of Edinburgh and Glasgow. A patient's electronic health record is typically fragmented across multiple digital systems, requiring clinicians to manually collate these disparate data sources to allow interpretation and decision-making for each clinical encounter. With the increasing complexity of cancer care, this data fragmentation has been shown to increase clinician workload, cognitive burden, and the risk of critical omissions, and this is well backed up in the literature. Large language models have shown promise for both extracting and summarising this information in controlled research settings. However, few studies have evaluated their performance on unstructured, real-world oncology data, which are often duplicative and inconsistent, whilst also being extremely domain-specific. In collaboration with DataLock and the Edinburgh Cancer Informatics team, a retrospective analysis was conducted on 10 selected breast cancer patients, spanning the care settings of neoadjuvant, adjuvant, and metastatic disease. The gold standard was created by a single oncology clinician. The method involved pre-processing the data from the various data sources. The data sources were cleaned to remove EHR-specific tags such as HTML and normalized to consistent date formats. Each data source was then routed to its own specific prompt. In each case, these prompts requested an output in a structured JSON format. The evaluation was performed by comparing the model outputs with that of the clinician gold standard. The model performance and results were evaluated using precision, recall, and F1 scores. Of the 539 clinical notes that were assessed, the macro F1 score was 79.6%. 75 radiology reports gave a macro F1 score of 88.4%, and of the 16 pathology reports assessed, the macro F1 score was 90.34%. Across all domains, the mean micro-average performance was a precision of 81.6%, recall of 87.8%, and an F1 score of 84.3%. The biggest impact on performance came from hallucinated side effects, particularly nausea and vomiting, which the LLM frequently generated in otherwise unremarkable notes. On reflection and discussion of this, the LLaMA 3.1 shows promise for clinical entity extraction. However, this evaluation demonstrates that hallucinations introduced by the model substantially limit its reliability in a clinical setting. Reducing such errors is essential before integration into clinical workflows can be considered. This study underscores the complexity of translating LLM research from the controlled or synthetic data environments into real-world clinical documentation. Future work will explore advanced prompting strategies and oncology-specific fine-tuning. This evaluation had several limitations. The first is that the gold standard was created by a single oncology clinician and therefore lacked the inter-annotator agreement. The dataset was also restricted to 10 patients, limiting diversity and generalizability. And as a single center retrospective study, documentation style and data quality may differ from those of other institutions. Thank you for listening.